This is a really exciting time for machine learning and biology. On the one hand, we're seeing innovations in machine learning algorithms that are enabling computers to perform tasks that we previously thought only a human could do. We're also seeing computers performing tasks that we haven't been able to solve before, like take the sequence of a protein and predict the structure of it. One of the promises of machine learning in biology is to assist in processing vast amounts of information at the level of individual cells to help us understand those rules of biology that give rise both to healthy systems and diversity across individuals, but also underpin disease.
I believe that we are at the beginning of a revolution for machine learning in biology. And that's why we are really excited about the NeurIPS competition this year. I think it's a unique opportunity to drive creativity in machine learning that will really enable significant advance in cellular biology.
Gene expression across the whole genome was observed something like 15 years ago, but only in a very small number of cells. Whereas nowadays, in the past decade, we can actually observe many hundreds, thousands even, up to tens of thousands of cells, and there's even some data sets going into the millions. That's why it makes it so interesting for machine learning techniques.
The DNA in the nucleus gets transcribed into RNA, which is the messenger molecule that exits the nucleus and then goes towards another organelle called the ribosome. And the ribosome translates this message into protein, and then protein is what actually has a function and is able to respond to any type of stimulus that the organism have to account for.
For this competition in this data set, we're measuring single-cell RNA sequencing, single-cell ATAC sequencing, and some protein measurements. Basically, we're looking at a data that's a big matrix. Each RNA species is a dimension, each location of DNA that's measured for accessibility is a dimension. And so this matrix gets to be very big. There can be 20, 30,000 or more features.
One of the most important components of developing machine learning models is access to high-quality training data. This is more complicated for biological systems because we don't have good ground truth. It's only recently that we've had access to molecular techniques that enable us to measure this ground truth, because it requires being able to observe multiple parts of the cell in the same cell at a huge scale. We brought together 4 partners at Yale, at Chan Zuckerberg Biohub, at Helmholtz in Munich, and at Celerity to generate a multimodal reference data set from 9 diverse donors, where from each of these donors, we took bone marrow mononuclear cells. These are cells from your bone marrow that give rise to the many different kinds of cells in your blood, including all of your immune cells.
It's really revolutionary that now we can have these different measures from the very same cell. For example, transcriptionally, the T cells look very similar to each other, but in fact around the body they perform very different and very distinct functions. That becomes relevant when they encounter pathogens like COVID or cancer.
The first task is broadly known as modality prediction. So that is predicting one modality from another. So for example, we would want to predict RNA sequencing from ATAC-seq or protein from RNA. And the idea here is that if you are good at understanding or capturing the state of the cell, you should be able to predict what the other modality is, because there's informational redundancy there.
I've always viewed the biotech industry as a kind of bridge between science and society. And the pandemic has shown the tremendous value that our industry is bringing to humanity at large. To those considering participating in this competition, you have a fantastic opportunity to directly impact human life.